conversation. So I want to thank you immensely for all your cooperation, dedication during my section in research methods. That's 803. So my colleague have completed my section. My colleague, Dr. Obuago, will I will encourage the course rep to approach him. If you need his contact, I will provide his contact so that he will continue from where I stop. He has a broader area to cover. So the earlier you contact him so that he will start in NS. Remember, next month, there may be exam for you next month. Already the center has arranged for a meeting. I think it should be virtual or on physical meeting on your research proposal. How you come up with your research proposal. I will encourage all of you to attend that proposal and make the best of best opportunity, the best opportunity of that particular program, so that you'll be able to firm up your research proposal and start up in NS. So we will be starting on, I understand also that Professor Mrs. Feichi has concluded her section in this course, environmental practice and environmental practice. So I will take the aspect of biodiversity in this particular discussion, this particular class, and also take off pollution control, pollution prevention, resource efficiency. I'll take that aspect. We share my slides now. So, and also want to inform you that all the topics I will have to take in this session have been uploaded in the LMS. So avail yourself the opportunity to download and read ahead of time, and keep your quiz on the note your questions. You can bring them up while the class is going on. Let me share my screen. That's coming up. Okay, so this is the slides, Introduction to Biodiversity Conservation. So you have had the section A. I will take the B section. We'll talk about the introduction, the objectives, the scope of application of diversity, the scoping for biodiversity during impact assessment study, and ecosystem services issues. We also talk about identifying and analyzing priority ecosystem services. Hope you can hear me clearly. So believe that we learn the concept of conservation, we define ecological habitats, and explain priority ecosystem services. So we believe that at the end of the section, the concept of biodiversity would have been known, and you have good knowledge of priorities of ecosystem services. So conserving biodiversity and maintaining ecosystem services and sustainable managing natural resources are fundamental to sustainable development. There is no way we can achieve sustainable development without feeding ourselves, without making sure that the environment is safe for every one of us, without making sure that we can have the fruits, we can have the oranges, we can have all our staple foods. We can go to the farm, grow our crops, and harvest them and eat. So there is no sustainable development without environmental sustainability. So it is very, very vital that we maintain the ecosystem services and also conserve our biodiversity. So biodiversity and healthy ecosystems strengthen our resilience to address environmental and social changes and shocks, including climate change impacts and disaster risk. So it's very important. Without the ecosystem, without the biodiversity, in fact, there is no way we will be able to combat climate change. Climate change is devastating the world because we have not paid enough attention to conserving biodiversity. It is very important to maintain and enhance the goods and services provided by biodiversity and ecosystems in order to secure livelihoods and provide food and have potable water and also maintain a healthy life. Biodiversity also enhance resilience, conserve threatened species and their habitats, and increase carbon storage and sequestration. That is what is so defining biodiversity. Like the name implies, we have biological and diversity. Is the variability among living organisms from all sources, including terrestrial, marine, and other aquatic ecosystems, and the ecological complexities of which they are parts. This includes diversity within species, between species and ecosystems. So within a landscape, biodiversity is defined as the variability, the number, the different types of organisms that inhabit that landscape. How many are they in number? How many are they the species or different types of species, within that particular landscape or ecosystem. So biodiversity features the suite of species. How many are they in clusters? How many are they in number in a natural communities, ecosystems, and ecosystem services, and ecological processes within the direct and indirect area of influence of a project. So when projects are cited or are proposed to be cited, the major impact, the major concern is always how will this project impact, have direct or indirect impact on existing ecological system or ecological process. Is it going to destroy it? Is it going to disrupt the normal cycle? Or what exactly is going to be the influence of that project? And that is where the direction we are coming as environmentalists, to make sure that project does not completely destroy our biodiversity and nature or ecosystems. Now, key biodiversity area, a globally important site that is large enough or sufficiently interconnected to support viable populations of the species for which it is important. Areas are selected based on the presence of globally threatened species, the presence of restricted range species. When we are looking at, please mute yourself, we can't be talking about this all the time. So when we are talking about restricted range species, there are organisms that are locked up, in an area that they can only be found in that particular vicinity or habitat. Attempts to remove them from there, they may not really exist again. So such range restricted species are very important in biodiversity study. So congregations of species that concentrate at particular sites during some stage in their life cycle, and the presence of biome restricted species assemblages. So these are all what we'll be looking at in some few days. Now, habitat. What is the habitat is all about? Is a terrestrial or freshwater or marine geographical unit or a way that supports assemblages of living organisms and their interactions with the non-living environment. So implicitly, a habitat is where organism is found. It could be in the upland, it could be in the freshwater, it could be a marine, but however, within that particular place or that particular unit, it has opportunity to live and survive and carry out the business of living, the business of looking for what to eat and how to survive. So where that organism is found is referred to as its habitat or its habitat. Because in most instances, habitat loss, degradation of or fragmentation represents the greatest threat to biodiversity. Of course, if somebody's house is demolished, means that the person becomes vulnerable to attack. The person can be robbed, the person can be killed. So likewise, species, likewise organisms, once their place of existence, their home is destroyed, of course, they become so vulnerable. So one of the drivers of biodiversity degradation or loss is habitat degradation or habitat loss, or a situation where they are fragmented into small sections. They will not have nesting places, they will not have places to hide, and so on and so forth. So much of the focus of biodiversity conservation actions is on maintaining or restoring suitable habitats. So if you want to restore biodiversity or conserve biodiversity, you make sure that the habitats are well preserved and maintained or have less disturbance. The objectives of the biodiversity conservation, one is to conserve plants and animals biodiversity to maintain and enhance the benefits of ecosystem services. When we are looking at ecosystem services, of course, guess you're not just hearing them. Ecosystem services are divided into four. We have the supportive services, we have the regulatory services, we have the cultural services, and we have the production services. So all these services put together maintain human life, try to improve on sustainable development and so on and so forth, to promote sustainable management and use of living natural resources. If we don't care for biodiversity, it will be difficult for us to exist. To ensure the fair and equitable sharing of the benefits from the utilization of genetic resources. Of course, if we destroy it, we may not be opportune to utilize our genetic resources. That is the beauty of it, to respect, preserve, maintain, and encourage knowledge and also innovations, practices of indigenous peoples and local communities relevant for the conservation and sustainable use of biodiversity. Of course, biodiversity is close to culture, is close to people's knowledge and practices. So destroying their forest reserve, destroying their shrines, destroying their river, where they go and make worship and so on and so forth. These are all attributes of biodiversity that should be conserved. Now, the scope of application of biodiversity projects in many different sectors can affect biodiversity. That one is sure, or even habitats. There is no way development project comes up that at least one tree will not be cut down or soil being excavated that will affect edaphic organisms, that will also affect boreal organisms. So definitely one or two organisms will lose its habitat. So what happens? So there is a wide range of project-related physical and biological impact that can affect biodiversity and habitats, including, for example, habitat conversion. If it's a wide, tropical forest, after the development, the area becomes grassland. So, which means the biodiversity, the habitat must have been converted. Interruption of important ecological processes such as species migrations, dispersal or pollination, degradation of habitat, quality from air, water pollution or temperature. There is a whole lot of changes once development project is coming up. So even if introduction of invasive alien species from exotic species from outside the area can also be introduced. And once that is done, it can outcompete existing organisms within the vicinity or even create problem, and as well as being vulnerable to fire and other stresses. So these are important factors, scope of application of biodiversity and what destroys biodiversity. Now, under many national legislative regimes, consultants are required to undertake a scoping process as a prelude to developing the environmental assessment. What do I mean by scoping? Scoping is when experts come together to brainstorm on ways to reduce the impact of that project on the biodiversity or other factors. Scoping try to identify all the impacts that project will have on the environment, on the immediate vicinity of the site where the project or the site of the project. So scoping can help to focus the environmental assessment activities towards addressing the most significant issues associated with a project. Scoping is an important step in effectively managing project impacts on biodiversity. Not only identifying the impact, scoping during project implementation will also identify the mitigation measures. As the impacts have been identified, it also behoves on the experts to go extra miles to identify how to manage that impact, reduce it to a manageable, manageable level. So project scoping undertaken by the consultant serves two major purposes: identifying potential environmental and social impacts of the project, as well as preparing the terms of reference for the environmental assessment, focusing on the significant issues. The team should review the project feasibility studies. Is the project very viable? Is it viable enough to have such impact? To the viability must override the impact the project will cause. Okay, or can we think of alternatives, either taking the project elsewhere or coming up with another plan for the project? And existing biodiversity and ecosystem services information available for the project, is that area a place of high biodiversity value? Is it a reserved place? Is it a forest reserve? Endangered species living within that area? You must assess to ascertain whether the place has a very high biodiversity area. That is the work of the consultant, that we assess the area of influence of that project before commencement of the project. So as an expert, a consultant, environmental consultant involved in EIA study, one of the key assignment, one of the key responsibilities you must have to assess or carry out is to assess the level of damage to the biodiversity within that area and suggest whether the project can be relocated. Also assess the project feasibility study, how economic visible visible is this project? Is it going to impact positively on the people or not? And now weigh the cost benefit analysis, the cost and benefit of you know of establishing that project vis-a-vis the impact it will have in the environment. Scoping should be participatory and involve consultants with stakeholders. Stakeholders are community members and other interested parties, the NGOs and other people within that particular community, including local communities that may be affected by the project, specialists with environmental and social expertise in the area, local government representatives, civil society organizations, and regulatory agencies. The scoping report should demonstrate that the client has efforts to achieve consensus with stakeholders on the approach to the environmental assessment terms of reference and the focus on the project. So scoping begins with summary document that has the following information. Project description. Before you embark on scoping that will involve assessment of biodiversity within that area, you must understand the extent of the project, what the project intends to achieve in terms of objective, scope, and so on and so forth, including the magnitude, timing, and the frequency of activities, project alternatives, project location, area of influence. You have to estimate the area of influence depending on the nature of the project, and preliminary if the project will influence people beyond area of influence, affect people living far off, beyond the state, beyond the country, and so on and so forth. Preliminary analysis of potential environmental and social impact. You have to assess, estimate and analyze the potential environmental and social impacts, including their spatial and temporal scale. What is the spread? What is the time and scale, and the description of the approach to public consultation. Now, the final scoping report should identify key biodiversity features. The consultants should be able to outline and document key biodiversity features. The presence of endangered species is a place with very high biodiversity value. Are there range restricted organisms? What are the nature of plants and animals? Are there economic trees present there? The scoping will come up with these facts, including the priority ecosystem services in the area of influence. Is the area, is it area of very important swampy area, mangrove, with very rich ecosystem that will involve reclamation and destruction of seafoods and so on and so forth, and should describe project potential impact and the risk for them. It should also identify relevant information gaps in knowledge, as well as any project-specific studies needed to understand potential impacts on biodiversity and ecosystem services. The scoping reports and terms of reference for the environmental assessment should define the need and scope of the biodiversity and ecosystem services baseline studies, including the proposed methodologies, sampling regimes, and should establish the focus for assessment and management of impacts on biodiversity and ecosystem services. In all this, the scoping exercise will address all this before the impact assessment study. Now, identifying and analyzing priority ecosystem services. We have defined what ecosystem services are, the services, the regulatory services. We know that most air, the clean water that we drink, the fresh air we breathe is also services of ecosystem. What ecosystem provides. That we plant our crops, we harvest them, there is pollination, there is recycling of nutrients. These are all ecosystem services. We also have places of worship, aesthetics, values. You have serene people visit parks, people go to areas, spring and war spring. These are all ecosystem services. They provide aesthetic values. You see this, area, serene area, you admire it. These are all ecosystem services. You go for picnic, you sit down and have a feel of nature. These are all ecosystem services. So identifying and analyzing priority ecosystem services. During scoping, the consultant should identify priority ecosystem services in the area of influence of the project that may be affected by the project or that may be important for attaining the development objectives of the project within a framework of sustainable development. This is very, very important. That is where your expertise comes into play as an environmentalist. The government may have economic objectives.
Possible you negotiate with the person who the place because someone don't quite negotiable.
Please, who is that? Please mute yourself. Now, what am I saying? The government may have economic intentions, but they are downplaying the environmental, impact of that project. It is you that will come up and, let the government know that this project, if implemented, will affect the ecosystem goods and services, and that will affect generations unborn, and so on and so forth. So you must make sure that you'll be able to identify priority ecosystem services within that particular area. Take, for instance, in a REM forest reserve that have stayed for many years, solving problem of carbon sequestration, providing clean air, and so on and so forth. Government decides to, cut down the trees to establish one project. Of course, if you have valued that ecosystem services is a very priority area, of course, you have the right to propose that this project should be relocated to somewhere else and now preserve these ecosystem services. Priority ecosystem services are defined as ecosystem processes, goods, and values that provide benefits to human communities and that may be significantly or adversely affected by the project or upon which the project has a significant dependence, so which should be avoided. Yes, ecosystem services may not be priced. That is a problem we have, but their services are invaluable. You're breathing, that means that place you are have offensive odor, you will not be able to stay there, or you pay heavily for each breath of air you take, clean air you take, because these are free gifts of nature. That's why it's completely abused. We do not reckon with it. We do not, think that is something that is very important. So it is important that we accord it the desired respect. The UNEP recognizes and safeguards the role of natural habitats in providing the ecological services required for sustainable human development. Provisioning benefits, I think I mentioned it, regulatory services, cultural services, and supporting services. And these are all ecosystem services: provisioning, regulatory, cultural, and supportive services. And in supporting the functional integrity of ecosystem generally, referred to as ecosystem services as recharging aquifers, sustaining fisheries, maintaining mangroves, or other ecosystems. Now, projects can affect the delivery of services to other beneficiaries. That one is sure, and also benefit from ecosystem services. The degradation of these services caused by a project can represent a legal, operational, financial, and reputational liability for clients. There is a prevailing trend towards the inclusion of ecosystem service values in decision making in the public and private sectors. But this is not manifested in individual project management. Understanding the cost and benefits of the relationship between project and ecosystem services is an important element in good decision making. But how prepared are we to make this decision over protecting the biodiversity, protecting the ecosystem services? Most times, the economic objective overrides the environmental objective. So we must strike this balance of understanding the cost of degrading or disrupting the ecosystem services. Most times, these services, once lost, cannot be reversed. They are irreversible and they are invaluable to us. Once it's lost, we cannot recover them. So that is why we must stand up and make sure that these services are protected and we have them and pass it on to our children. Now, the analysis and description of ecosystem services can be undertaken through qualitative, quantitative, and monetary approaches that provide complementary information. Qualitative reviews identify the range and extent of services as well as the beneficiaries and users, and they document flows of services from source to sink. So it's beyond the scope of this study when we look at environmental economics. I think we'll have to do that course. Is this this semester or next semester? I don't know the option that does that course. So that is when you begin to quantify the externalities, environmental externalities or internalities of these ecosystem services and how they are priced and how you value them. Now, where a project is likely to affect the delivery of ecosystem services to beneficiaries or directly depends on ecosystem services, the consultant should undertake a qualitative review of ecosystem services during scoping. This should be participatory and occur in parallel with the scoping process for a project. It should involve the beneficiaries, that is the community members, indigenous people, as well as the relevant environmental and social technical specialists. They should be present. Let's value it, because it's not an easy when the community is desirous of the project. You're telling them that, look, this area, this project is going to be sited, is not good. We are thinking of relocating it. So you come up with a superior argument concerning the preservation and quantify the damage, the level of damage, and the services this ecosystem, provides. Assuming that that is only portion of land the community depend on for their farming, and it's being taken away from them. How do they survive in terms of growing their crops and all that? So you should come up with a better analysis, the qualitative review of the services the ecosystem provides before commencing the project. Now, the qualitative review of ecosystem services should document the sources, flows, and beneficiaries, including the project for ecosystem services that are relevant in the project's area of direct or indirect influence. It should also determine the beneficiaries' degree of dependence on the services. I've also explained this. Describe the recent trends in delivery of the service, including determining the drivers of change within that particular ecosystem, what can drive the change, what can destroy it, what can affect it negatively, and describe the potential changes and their magnitude that may occur due to the project. Those of you in risk assessment option, these are, you continue to come across this in field studies. If you have been involved in EIA studies or have been involved in EIA review meetings. So these are critical areas of argument, concerning what should be done, what is the degree of dependence of the services, and describe, and also how the community members depend on those services before justifying that the project should be sited in those areas. It takes a genuine and diligent study to identify the biodiversity concerns and decide that these biodiversity are not relevant. It can be avoided, or the impact may not be grievous to the community members. So, and describe the potential changes and their magnitude that may occur due to the project. The qualitative review should take into account non-local beneficiaries of services and should include non-instrumental and non-material services. The ecosystem services review report should include this information, as well as identify the key social, operational, financial, and regulatory, and reputational impacts on and the risk to ecosystem services that are related to the project. The consultant should also apply the mitigation hierarchy and identify actions required to minimize the impacts of the project and identify the ecosystem services. When we are talking about the mitigation hierarchy, it's a four-stage process where you have to, for any project, you have to think of sustainable means of implementing that project. You must think of sustainable means of implementing that project, avoid the impact, avoid such actions that will bring about adverse effects to the environment, and now think of how you can reduce that impact, and now provide minimized impact, or you can also offset. So these are four mitigation hierarchy: sustainable approach to the implementation of the project, identify the impact, reduce the impact, and also think of offsetting it, but can pay compensation where avoidance is not possible. Now, biodiversity is crucial in the delivery of ecosystem services. Very, very important. We must preserve our biodiversity. There's no two ways about it. They play a major role in our existence. And there's no way sustainable development can be achieved without protecting the biodiversity. So biodiversity consideration is vital in environmental assessment for development projects, and should be carefully carried to ensure sustainability of ecosystem services during and after project implementation. So it's important that biodiversity are given very utmost priority during project implementation. And experts should be able to provide a real-time assessment of the biodiversity condition, the ecosystem services that is the area that is being provided before deciding on what to do, whether the project will be sited in that area or not. So thanks for listening. I will quickly go over to the next slide, but before that, I will take your questions on this introductory. The introductory has dwelt on the initial engagement during impact assessment study, the scoping, and why it's necessary to evaluate, brainstorm, and decide on the nature of the particular area in terms of the biodiversity and ecosystem services the area provides, making sure that the project will not diminish or destroy the ecosystem services that particular vicinity biodiversity or that area provides to the community members. We must strike the balance, assess the cost and the benefits on the ecosystem goods and services before that economic project is cited. I will be glad to take your few questions before I will share the next slide. Thank you. Any question?
Good evening. Hello?
Okay, good evening.
Yes.
Okay, go ahead.
How do you gather this data from biodiversity? Is it a primary information, or do we gather it on information that has already been existing in the environment? For example, studies that have
Okay.
been done to know the kind of organisms or animals that are involved in the environment.
Okay, thank you very much for that question. The next slide, we are going to deal on sampling and gathering of the methodology involved in considering ecosystem biodiversity. There are a whole lot of techniques that we can adopt to assess the biodiversity. To answer your questions, you must go to the field, consider the plants, not only the field, it can also rely on the literature, previous work done in that area, and also do a bit of ground truthing to know the existing species of plants, and also know exactly if you, as an ecologist or a vegetation expert, if you go to an area, you should be able to assess the level of services that ecosystem provides and be able to evaluate the need, and as well as weigh the cost on the loss of that biodiversity to the project. So these are things we need to do. So in the course of this lecture, the next couple of weeks, we should be able to understand the need for ecosystem and the way we go about it. Studying it can be laborious, but it was the tax, it was the effort works, because once we destroy biodiversity, destroy the environment, we all finish because of our quest for economic development. So we also need to preserve the ecosystem. We need to preserve the ecosystem services that we enjoy. So any other question before I will share my next slide? If there is no more question, can I unshare this and share the next slide?
No,
Yes,
my
Prof.
girlfriend, I go tell you, I see her, my one of my friends.
Are you asking a question, that young guy, or are you?
Just making my.
Okay.
Move ahead. Move ahead.
Okay, good. Thank you very much. Let me upload the next slide.
Other one, downstairs. Downstairs. Go to that.
So we have the biodiversity assessment techniques. This is in line with the question our sister asked. So we look at the assessment techniques, and it's good. These are things we take to the field. If you understand the techniques and you get involved in EIA studies, and you have a knowledge of biodiversity study, you take the responsibility. So we have the introduction. We will discuss the dominance index to assess species diversity, Markleaf richness index. We have Shannon-Winner index. These are all authorities that have come up with models that will assess species diversity. Bonnerin index, log series index, species evenness index, and percentage minority index. These are all indices to assess biodiversity. Now, at the end of the objective is to explain the dominance index of biodiversity, to explain the Markleaf richness index, to explain the Shannon-Weaver index, to explain the Burling index, and explain the log series index, and so on and so forth, percentage minority index. To believe that at the end of the day, this index, this assessment index, we understand it and apply it in our field biodiversity assessment. Now, biodiversity assessment is the first step in protecting the complete range of morphological and genetic diversity of species on Earth. I said is the first step, in protecting, because if you don't assess something, you will not know whether it needs help or not. You must go in and know what it looks like, what the nature is, what the dangers are, and what the level of damage. Otherwise, you will not protect. You will not have any idea what the situation is. Range of morphological and genetic diversity of species on Earth, and enriching the goals of conservation biology. Assessment begins with identifying organisms that make up biological communities and understanding evolutionary histories. Scientific advancement in molecular sequencing can help clarify and provide support for identifications. Sometimes identification is done at molecular level. DNA fingerprinting and sequencing, molecular sequencing, will actually identify and separate biological communities. So we cannot exhaust all in this study, but however, we are going to, our main target is to do the assessment, know with the techniques that are used. We may not really go deep into identifying. These are the botanists and plant taxonomists who handle that. Now, massively parallel DNA sequencing technologies are being used to sequence complete genomes of model organisms. However, this resource has not been fully used for species identification. Like I'm going to do heavily on species identification, that is just for sake of mentioning that we need to identify organisms, but our own is to provide assessment tools that will quantify the biodiversity present in that particular community. And groups of plant scientists, chloroplast markers, and genomic identifiers. Yet, nearly as many studies have reported that the proposed regions are uninformative in some plant groups. So it's inexhaustive identifying organisms. So conservation planning begins with a comprehensive evaluation of regional biodiversity, knowledge of organisms' life history, species richness. Species richness, how many species are there? How many types of plants are there? Endemism, how many of them are endemic in that area? There's a guy disturbing us. Please call the guy's attention to mute. There are, yeah, if you go to, if you can say go to Benin, that is where you will find this species of plants. They are rarely found in this area. So endemism, they're endemic, some species, some animals are endemic in certain areas. That is life history. These monkeys are not found in Imo State. They are found in Cross River State. That is where they are found. Reality is very rare. These organisms cannot be found in the tropical climate. They are always in the desert. They are rare. Range of morphological and genetic variability. If you find these plants in south, they have broad leaf, but the same plant can be found in the north with vein leaves. So the range of morphology, still the same species, maybe due to environmental influence, they tend to change their morphological features to adapt to the stress conditions, to adapt the condition within that particular community. Even animals can change their color, to adapt to certain, environment, maybe to reduce their vulnerability, to escape predators, and so on and so forth. So what are the range? These are histories that is needed to understand organisms. It's a life history, species richness, that particular area, and also an evolutionary histories are all parts of a biodiversity assessment. But the necessary first step is identification of the organisms themselves. These organisms, what do they do? What are they? What kind of organism is this? How does it feed, and so on and so forth? You identify the organism. It depends. It lives on this, and so several calls have been made in the last 15 years for systematics to contribute to conservation and biodiversity assessment and to make taxonomic data more accessible to a larger audience. So by way of introduction, we have the newest DNA sequencing method called massively parallel or next generation technologies capable of delivering more data at lower cost and with less pre-sequencing laboratory preparation than traditional Sanger sequencing, which has been the standard for over 30 years. So this is, we're not going to dwell so much on the identification and the molecular level in this our study. Now, this is the flow chart of biodiversity assessment. To have the plant survey and sample collection, you go to the field. You have the classical taxonomic, you can approach it, classical taxonomic and genomic sequencing, and to know the gene at the gene level or species level, where your collection sample you've collected is from. You have to now identify the species and move down to understand the biodiversity. You can talk about the phenologic diversity, endemism, rarity, morphological variation, species, richness, and life history. So by the time you understand the diversity, biodiversity of that organism, we now begin to talk about these other areas. So talk about the phylogenetic diversity, phylogenetic diversity, that is the physical appearance and the morphological variation, and so on and so forth. Now, this index, dominance index, this is an abundance index that measures the probability that two samples taken as random from the data set represent the same species. You move into a community of plants, you randomly take two samples. At the end of the day, those samples belong to the same species. Belongs to the species. What does it tell you? That particular sample you may consider it to be dominant, within that plant species. Maybe Chromonella odorata, Wolowo. There are so many plants that are predominant in that area. So if you close your eyes and sample two, if they belong to, feel that you can categorically say that this sample is dominant in that area. They are higher in number than other species. The mean proportional abundance of a species increases with decreasing number of other species, or with increasing number of that species. Hence, the index will be small when diversity is high, or large when diversity is low. Of course, it varies, the value ranges from 0 to 1. So with increasing number of that species, the index will be small. That is, then we have, there is no, it cannot underpin a species that is actually in dominance. That is, the diversity is very, very high. But once the value is high, then it's low, that means there is high dominance. One organism must be dominating in that particular community. Now, species richness stroke abundance. Abundance of species is evaluated by counting the number of individuals of a species in each Whittaker transect. That is, transect is one of instruments we use in vegetation study. Within that particular area, you will now count the number of organisms, the number of species. Now, abundance of species is evaluated by counting the number of individuals. If we have 40 different individuals there, and that 40 individuals, 80% belongs to one species. You understand? So the abundance of species there, the richness is very low. Family abundance is evaluated by counting the number of individuals of species belonging to a family. Now, we also have a Mark-Leaf richness index. It is a measure of species richness in a community, often used in ecological studies to assess biodiversity. It focuses on the number of species in a sample relative to the total number of individuals, and provides an easy way to compare species richness across different environments or time periods. Please, you do not have to confuse yourself with the word individuals and species. Individuals, individual refers to stands, while species refers to type of plant. Okay, so we may have 40 individuals, but we may have 10 different species. You understand, making up 40 individuals, and provides the number of species richness. So in species richness, we look at what? Number of individuals. This index is a measure of total number of species present for a given number of individuals. For the same number of individuals, the value will be high if it contains more number of species, and vice versa. If it contains more number of species and vice versa, the values can come more than one, with higher values indicating high richness. The index is strongly influenced by sampling effort and sample size. This index shows the richness of species in a community or ecosystem. It has high discriminating ability and is sensitive to sample size. It is a measure of the number of species, measure of number of species present for a given number of individuals. However, it is weighed towards species richness values. Values can be greater than 1. So the higher the value, the more richness. Wow. The Mark-Leaf richness index is defined as D 1 over the Wonder why this thing is removing, but I guess it should be in your notes, the one I uploaded, the model for the Markleaf index. I don't know why LMS always remove all these model equations. So where the total number of species in the sample, species richness, total number of species in the sample, that is species richness, and total number of individuals in the sample, and lean is the natural logarithm. Of course, we have an example here talking about calculation of species richness using the Markleaf model. We have the species, we have A, B, C, D. Species 1 has 10 individuals. Species B has 15 individuals. Species C has 20 individuals. Then D has 5 individuals. Total, we have 50 individuals and have 4 species in this community, in this plant community. So in calculating the richness, we have identified the parameters. We have X, the species, number of species is 4, number of individual species, that's is total number of individuals is 40. So the Markleaf index is number of species minus 1 over lean of the total number of individuals. So once we substitute that, we have 4 minus 1 lean of over lean of 50. So we have 3.9 lean of 50. By the time we resolve that, we have 0.767 as the index. So we can interpret this as Markleaf index for this community 0.767. So this value reflects the species richness of the community, standardized by the total number of individuals. Larger values of D indicates greater species richness. Like we say that it can be greater than 1. So if it's higher than 1 means that it has species richness. You can describe that community as having very high number of species. Now we also have Shannon-Wiener index. Shannon-Wiener index is the most popular index to measure species diversity, and most textbooks define it as the index. It represents a combination of richness and abundance. Combination of richness and abundance. It quantifies the uncertainty in predicting the species identity of an individual when taken at random from a data set. If there is only one species in the data set, then Shannon entropy exactly equals 0. There is no uncertainty in predicting the species. Uncertainty, there is no uncertainty. That means that particular community is dominated by one species. So the diversity is quite low. This index increases with increasing diversity and evenness in the data set. This indeed assumes that individuals are randomly sampled from an independently large population. The index also assumes that all the species are represented in the sample. Logarithm is often used for calculating this diversity index, but any log base may be used. The value of Shannon diversity is usually found to fall between 1.5 and 3.5, and only rarely it surpasses 4.6. So if you are working on determining any species diversity and it says 4.5, you know that you're very wrong. You're very wrong. You cannot be correct. So it's important that we know how most of these models work and function. Now, another example of Shannon and Winner species. This is the model minus summation proportion I, lean of proportion I, where H is the Shannon's index, lean pi is the proportion of individuals belonging to species I, the proportion of individuals, proportion of the total, the proportion of the species, assuming that the species is 10 in number, the total, the proportional over the total, the number of that is species all over the total. That gives you the proportion. Look, take the example I have here. We have a hypothetical example from an ecological community. We have A, species A, B, C, D. This is similar one we talked about in the market leaf. We have 10. We have B, species B, 15, species C, 20. Then D is 5. The total is 15. To get the proportion of I, we have 10 divided by the total number. We have 0.2, 15 divided by the total number. That gives us PI, the proportion of these individuals in this community. Now, we move ahead to get the lean of PI. The lean of PI is lean 0.2 give us minus 1.606, this minus 1.2, and so on and so forth. Then going by the question, we need to do summation. We need to add the proportion, not add. We have to multiply the proportion and the lean and now sum up. So by the time we multiply lean 0.2 by minus 1.6, we will get 0.322, 0.3 multiplied by minus 1204, we get 0.2. At the end of the day, we will now do summation. We add up 0.322 plus 0.361 plus 0.366 plus this. Once we have done that, we will get our Shannon Winner diversity index. Here we have 1.279. The interpretation means the Shannon index for this community is 1.29, which reflects moderate diversity. Higher values of indicates greater diversity, considering both species richness and evenness, which means this particular forest, this particular community is moderately diverse. In fact, by looking at these numbers, you cannot underpin a particular species that is dominating that. So you will see that it's very dominant. Species are very rich. This one 10 in number, this one is 20, this one is 15. This A, B, C are all competing in this particular community. The only one that is very low in number is very D. So the richness here a situation, please let's mute ourselves, please. Situation where, for instance, A becomes 40 and the rest are 2, 2, 2, 2. Of course, you know that this is less diverse. A is actually dominating, so you don't consider it to be have a high diversity. Now, borrowing index. The borrowing index is a measure used in ecology and biodiversity studies to assess the diversity of a community, a biological community. It quantifies the distribution of species in a given sample. It quantifies the distribution of species in a given sample, accounting for both the abundance of each species and the total number of species. Borrowing index is particularly useful when dealing with data collected from finite samples and when species abundance are known precisely. It is used instead of the Shannon index when diversity of non-random samples or collections is being estimated. It is used instead of the Shannon index when diversity of non-random samples on collections is being estimated. So this burning index relies on non-random samples, works on random, whereas the Shannon index deals on random samples. For instance, fishes collected by attracting them towards light create bias samples, since not all the fishes are attracted by the light. So burning index is used here to calculate the diversity of fishes collected by gears that use light for fishing. Thus, this index serves the purpose of purposive or bias sampling. These are things you need to know. Now, the formula for this is not shown here, but it will be in your note. I don't know why this model is not seen here, but if you can, somebody can confirm from the, let me quickly check the note. I think it should be there. It should be there in the notes.
Okay.
Okay, is there in the note? Please, you is there when you upload your note, you will see it. So now it's there in your notes. So it's a very you look at the formula. The N there is the total number of individuals in the sample, and is the number of individuals of the height species. Height is the total number of species in the sample. Length, the natural logarithm, and the factorial. So when you look at the model, you can now substitute. Why the Shannon index is also a measure of diversity. It assumes random sampling from an infinite population. So Shannon index deals with random samples, whereas Burning index deals with non-random sample from finite samples. Consequently, Burning index is often preferred when exact counts of individuals are available. So you can use that. Log series index. The log series index is a statistical model used to describe species abundance distributions in ecological communities. It provides a way to quantify biodiversity by characterizing the distribution of species with varying abundance in a sample or population. This is widely used index to compare the diversity in similar habitats. It has good discriminant ability and the fact that it is not influenced by sample size. Long sampling periods and large sample sizes are required for computing this index. The log series is based on the assumption that the number of species with X individuals follows a decreasing distribution. It models the likelihood that most species in a community will have few individuals, while only a few species will have many. The model here is also not captured. You also find it in your test, in your notes. That's already in the LMS. You download it, you will get it. It's just there. The model, you look at the model for this log series index. Now, there's another model again, the species evenness is also an index to measure biodiversity. Evenness index is also an important component of the diversity indices. This expresses how evenly the individuals are distributed among the different species. Evenness index measures the evenness in proportional abundance among the types, and the value ranges from 0 to 1. It is estimated based on Shannon index and the total number of species. The values near to 1 will indicate high evenness among all the species. Shannon equitability index, will have it as EH equals Shannon wither all over ln of X. where H is the Shannon index, S is the total number of species. So the index can be measured and determined. Now we have the percentage similarity index. Now we use this to compare communities, to compare the species composition between seasons or even communities. This index reflects the similarity in percentage abundance or different species between seasons. Also can be used to compare species within two different communities. The PSIs close to 100 indicates the highest similarity, compare how similar communities are. Now, Pamanova is called permutational analysis of variance, and this is a statistical test used to test the significant difference between various sizes of a species or a species assemblage. Algorithm and SIMPA. These are the acronyms used in this particular Pamanova studies to test the spatiotemporal changes in species assemblages. A non-parametric analysis known as the permutation-based one-way analysis of similarity. Algorithm is used to test the hypothesis that no changes in community structure were observed between sites, time periods, different habitats. Aerostatic value for pairwise comparisons provided by algorithm are used to determine dissimilarities between these groups. So, which means this can be used to assess, of course, this is a statistical application that can be used to assess two communities to see whether there is change, spatial-temporal changes within the assemblages of that community. So using this aerostatistics value close to 1 indicates very different composition, while values close to 0 indicate difference in composition. So while in simpler, the standard deviation of the Brachytis dissimilarity matrix attributed to a species is used to compare the average contribution of that species to the dissimilarity in a particular site or session that is involved. So I will be glad to take your questions from these slides, or even if you remember another question for the other slide, you can also ask. These are a few of the techniques we can use to do vegetation study, apart from the qualitative assessment. This is quantitative assessment. We also can do qualitative assessment where you move into a landscape, a vegetation area, and assess how that particular project we have on that particular ecosystem and estimate the ecosystem goods and services that it provides. What is the level of influence of that project? Will it affect an area with very high biodiversity value in terms of conserving the species richness of that particular area? So now this method, this second slides I explain now are the quantitative determinations, how you can count the number of species, quantify them. Is this landscape, does it have very high diversity? What about the evenness? What about the species diversity? What about the species richness and abundance? So these models we have explained there can be used. You can use any of them depending on the one you are familiar with, depending on the one that you can use to quantify the species richness or the species diversity of that particular area. They are all important. They are important data. They are important values that you obtain to support your argument about that particular species. Sorry, just a minute, I'm coming. Hello, can you hear me? Hello?
Yes,
Yes, sir,
Prof,
we
I
can
can
hear
hear
you.
you.
Yeah, I'm
We
very
can hear you,
sorry.
sir.
I had to step out to attend to somebody. So if you have any question, you ask, and it's also good that you read the notes. If you have any area confusing you, you draw my attention to it, please, because these are practical things you need to do in the field. These are practical things you need to do in the field. You can't avoid it if you really want to carry out ecological study. If you want to carry out EIA study or ecological study in the field, you need to do all this. So any question? I know you have another class now. This is to 6. After 6 o'clock, do you have another class today?
Hello, Prof. Yes, we have another
Yes,
class
go
today.
ahead.
We have another
It's
class.
okay. Now, you may, my own is, I know that you may not get all your questions now, but I'm at your service. Read through the material and ask questions. If possible, get any textbook, biodiversity textbook. All these models are there. The Shannon and Weaver index, they are popular. Market leaf index, they are popular. Dominance index, they are popular. These are tools for ecological study. These are tools for biodiversity study. For quantitative biodiversity study, you can as well and do a qualitative study also based on your experience to evaluate the ecosystem services that community provides, and now give honest advice on whether the project will proceed or not. So I don't know if you don't have question. I may say we'll call it a day. And perhaps meet next week. And yeah, we'll cover too, our normal tradition that may be text attached to this quiz, a small quiz of maybe 10 questions or more that you need to answer that will form part of your CA. So the quiz may be available. Today is what? Today is Monday. Maybe from Wednesday, the quiz may be available. So you don't forget, I would have given you time to read through the material so that you understand and you answer the question. So Wednesday, Thursday, the quiz will be available. So be rest assured by the week, before the week runs out, you will take the quiz for these 2 slides. Thank you very much. I also want to thank you for your performance in the research method. Some of you did very well. You scored 50 over 50. Just few that could not get any score, and I'm not happy about that. Performance is not good enough. So those people that could not have any score in that last quiz, you have to wake up. You have to approach some of your colleagues to take you through that particular lecture. My colleague will be coming very soon, so you reach out to him. Tony, please call Dr. Boago. Don't relax. He's a HOD. He has so many things around his neck. So please follow him up. I'm sure that he comes. His aspect will help you. Somebody is raising his hand. Adedoyin Oluwafemi. So you have the microphone.
Good evening, sir.
Good evening.
Yes, sir. Good evening, sir. Thank you
Go
for your
ahead.
time.
Go ahead.
I'm so sorry.
Yeah.
You're the one that took us on 803.
Yes.
The last quiz, I actually missed it, and it wasn't intentional.
No, that's not what we are
because
talking
I
about.
was-
No, hold on. Let me cut it short. That's not what we are talking here, please. That is gone. We are talking about
painful,
801.
seriously.
Yeah.
It was painful, and I don't know if there's any help that can be offered, please.
Bernard, do you want to say something? Bennett, Uzo here.
No,
So
sir.
if there is no questions, let me allow you to prepare for your next class, and let me also prepare for my next class. Thank you very much. Thank you.